Earlier this month, when we had our Art and Science Cafe, but it's wonderful to welcome everyone to our first official Science Cafe of the season. We're really happy to be with you in person, and also happy to have a wonderful online audience today. So thanks to all of you for tuning in. And I'm sure you don't need reminders, but if you have any questions or any comments, if the sound quality is not great for you online, please put that in the chat. And we have some folks here that are monitoring that for us today. So we'll try to make sure that your experience is as good as the one that we're having here in person. So today we're talking about organoids. And without the scientists in the audience, I'm really curious how many people here have heard of organoids before. Yeah, I've heard of them a little bit. Curious to know what they are, what they mean, right? Me too. I didn't really understand either until I learned a lot about the nucleus. So I want to quickly give you a bit of an overview of why organoids are important, what they are helping us accomplish here at the MDI Biolab, as well as in scientific enterprise worldwide, because they're an exciting new tool. It's kind of taken the research community by storm, and people are really excited about the potential for using this technique or tool in a way that will help us better understand disease, that may help us eventually develop new organs, which is really what we're going to talk about tonight with some of our.
No.
Oh, yeah,
No.
Okay.
So we are just going to talk a bit about organoids, what they mean to the scientific enterprise, and specifically
hard to hear.
The MDI biological laboratory in that regard. And you've heard us talk a bit about kidney disease before, right? It's hard to say the word MDI biological lab without using the phrase kidney disease, because we do have a long history in understanding kidney health, but also what happens when we have a disease state in the kidney. And so, you know, you may not realize it, but 1 in 7 Americans have kidney disease. So, you know, just in this audience, there's a pretty good chance that there are some folks who are affected by that. The interesting thing, though, is that 9 out of 10 people that have it don't realize that they have it. And that's the real danger with kidney disease. So that presents a number of challenges, as you can imagine. Often people aren't diagnosed until they are in the final stages of kidney disease. So that makes treating it pretty difficult. And that's part of what we are trying to address here, is to, number one, find some earlier indicators of kidney disease, but also to come up with better treatments, because today you're pretty much limited when you're in the final stages of kidney disease to either having dialysis, which is like, Dr. Haller can tell you more about that, but very time consuming and a pretty big disruption in your life, or transplant. And I think right now, most people wait between 3 and 5 years to receive a kidney transplant. So you can begin to see the enormity of the problem. And so the scientific community in general is really working hard to come up with better solutions to address this kind of thing. And that's part of what we want to share with you this evening. So without further ado, I do want to introduce Fred Bever, my colleague Fred Bever, who's going to kind of moderate our discussion this evening, and he's going to introduce our panelists to you. So Fred, take it away.
Thanks, Jerry. So first, just want to check that we are indeed, the people online can hear us. I am the Chief Communications Officer here at the lab, which is a wonderful job. And I think most of you are fairly familiar with Dr. Haller, graduate of the Free University of Berlin, did his postdoctoral work at Yale, has had a distinguished career, 700 articles, 6 patents. And of course, after many years as a director of the Hanover, is it the Institute of Nephrology? Is that how we should pronounce it? The Department of. He, in 2018, became our president after many years, decades, as a visiting scientist here. But you might not know Cory Johnson quite as well, although he's a local boy. He grew up in Camden, and he did have a life before science. He was an outdoor educator. Fair to say a ski bum.
Oh, absolutely.
But then went back to school all the way through at the University of Maine. Is that right? Yeah, from BA or BS, I presume, through his doctorate, which he received just last year.
Yes.
From the Graduate School of Biomedical Sciences. And so now you're doing your postdoctoral research here, and you've chosen Dr. Haller's lab. You just returned from Germany. And what were you studying there?
I was learning how to build kidneys.
Learning how to build, and that's what this is really all about. You know, one way you can think about organoids is kind of like lab-grown tissue, but kidneys are really complex. So, Dr. Haller, why kidneys and why organoids? Why is this work important to you? Why is it important to all of us?
Well, I mean, Cherry already mentioned one important point, and this is that we need kidney. You know, kidney doctors, we are the only one who can replace organs once they've failed. We can do dialysis, we can do transplantation, but the holy grail is really to make a new kidney. And the pressure is very high. Cherry mentioned all these patients out there, and they don't know. The US and all Western countries are spending more money on dialysis and transplantation for kidney failure than for any other disease. So this is one reason. And the other reason is because MDI-BL, we are kidney. I mean, we have been kidney for 100 years. We have learned here how the kidney functions. We have understood all the complicated mechanisms. We have used the animals here. And it's not only the two of us or our whole research group working on that, it's also Ian Drummond. You know, when we recruited Ian 4 years ago from Harvard, he is actually the most senior scientist for a program at the NIH, which is called Rebuilding a Kidney. So there are all the researchers from the US coming together, and there is lots of discussions how to do all that. So this is why we are doing kidney. And one more remark, it's an international problem. So we all have to work together. I mean, obviously, it's also competitive. We want to beat the others, but not by inventing it ourselves, but winning in our field, which is vascularizing the kidney. We want to be the first to get blood vessels into the kidney in a coordinated fashion. That's what we're going to do. And Cory just was in the Netherlands and met Ton Rabeling, is also a lot of money. Ton Rabeling, one research group, has 250 million euros in order to solve the problem in the Netherlands. And this gives you an idea how much money is involved and how many research groups. So we have to work very hard to make Cory famous and to make ourselves famous in order to be the first to get blood vessels into the kidney.
And Cory, I'm curious, when I was talking with you about this a few weeks ago, we talked about both the need for donor organs and the need to not need donors, but actually be able to create organs in the lab. But for you, it wasn't just about that results, right? There's something about the science itself, which I'm just curious, like, besides that, what makes this interesting for you?
Well, so I'm a basic biologist. I like to look at things at a cellular and molecular level. And so I think the organoid in particular is a really interesting tool to be able to do that and see how these cells can kind of self-organize into tissues and then eventually organs that are more complex than, you know, we could really fathom with all the different levels of proteins and things like that. So I think being able to ask questions about that organization is really interesting.
And so it's sort of the joy of basic research,
Yeah.
right? This is a pretty good place to do it. Yeah.
It's a great place. I mean, we're not only using, you know, the top tier tools, microscopy and techniques here, but we also get to do it where my office is 100 feet away from the ocean. So I think it's the ideal place.
And access to the international
Oh,
cadre
absolutely.
of experts.
It's one of, at least from my perspective, being at UMaine, where there's a lot of international students that come, I think this is the most dense area of international contributions that are happening in the state that I at least have been a part of.
Okay, so tell us how you do it now. How do you build a new kidney? We call this making kidneys from scratch,
Yeah.
but we're the scratch, right? It's
Yeah.
human stem cells.
Yeah, so we use, it's a commercially available stem cell line, but we use stem cells that were taken from a blood sample, then turned into stem cells. Sorry, they were cells taken from a blood sample, turned into stem cells. And then we feed them different signals, different molecules that provide them the starting material for how they can self-organize themselves.
Well, take me back a second there, though. How do you convert a blood cell into a stem cell? What do you
Yeah,
do?
that's a good question. So in 2009, might have been 2008, Yamanaka, who's a Japanese researcher, discovered 4 essential proteins that regulate gene expression, and found that if you induce their expression in human skin cells, that you can turn them into stem cell-like populations of cells that have the capacity to turn into basically any cell type.
If I may add to that, I mean, everybody understands, you know, this shows you 2 things. One, it's very complicated. I mean, nobody would have thought in 2005 that it would be only 4 factors you need to make a stem cell out of your cell. I mean, Cory could go out now and we could scrape your skin and we get a couple of cells from you and we can make stem cells from you individually just by these 4 factors. Yamanaki got the Nobel Prize within a couple of years. So we are hoping for doing something like Yamanaki here. At the same time, then it just galvanized the whole field for the next 10 years. And this is the promise that we can take your cell and we can turn them, and not only us, but other labs as well, we can turn them into stem cells. And then if we are nice to these stem cells, and if we are very convincing, eventually we'll grow your own kidney from your own stem cell.
I've been curious about this. Does this mean that the need for embryonic stem cells is reduced or obviated, or?
Yes, no embryonic stem cells anymore. You may remember, I mean, this big discussion about the ethical problems of using embryonic stem cells. This was, the Yamanaki discovery was really groundbreaking because now we can take cells from individuals and turn them into stem cells in a dish.
So you can, those 4 factors that he identified them in embryos, right? But now you can make them outside without the
Yes.
embryo.
And now the problem, so we can actually, and people are using this worldwide, that for instance, when we have a patient with a genetic disease, when we get the stem cells from this patient, we can then mimic the disease, and then we can, for instance, use different therapeutics and test them. In Europe, for instance, and in the US as well, when we have young children with cystic fibrosis, all of them have their organoids somewhere stored. And before they are treated with a new compound, the compounds or the combination of the compounds are tested on these organoids. This tells you how powerful the tool is. These organoids are still relatively easy. Ours are more complicated, but it has been a revolution.
Cory, I think you made up some slides. I'm wondering if you could tell us a little bit more about the making of an organoid. I mean,
Sure.
you know, I don't know, do you take a little lattice of straws and drop material on them?
Yeah,
Can you
exactly.
help us understand that?
No, so we start with a dish with stem cells, right? And they're just kind of growing, proliferating, and then when we decide that we're ready and they're ready to move on to the next steps, we change out their nutrients and the factors that they need in order to do that. So I don't know, Roy, if you want to put up the first slide, and we can talk a little bit about what that looks like at the end of it. So hopefully the video plays, but this is kind of a rough example of what an organoid looks like at the end of culturing it, at least in what we're doing right now. So everything that you see in pink and blue or cyan are parts of the functional kidney. So the nephron, which is what ultimately water gets reabsorbed through the kidney and salts get exchanged, etc. But what's really cool is what we were talking about earlier with vascularizing these tissues, is that we have these green blood vessels developing throughout the culture, which is kind of what we're trying to do. If you look really closely, and I don't know if the video is playing.
No, it's
Oh, great. You can see a slight separation between those two things. If you look close between the green and the other colors, the green is on top, but it hasn't really infiltrated into these organoids yet. And so ultimately what we're trying to do is to get that to happen. Yeah.
Now, Cory is too modest. At the moment, if I may, there is nobody on the planet who can do better blood vessels, the green stuff around it, than us. So we have found a couple of factors, not like the Manaki factors, how we can introduce the transcription factors in these endothelial cells. So that's a big breakthrough. And now the next step, as Cory mentioned, is now we have to go inside. And you can imagine, this is wonderful research, but this is like growing an embryo. So what we are actually trying to do is we are trying to mimic what's happening in normal embryos, getting the blood vessels into it, and not like in a tumor, but in a coordinated fashion so that it really goes where we want them to go to.
Dr. Haller, in a previous conversation, referred to this as the miracle of human architecture that we're trying to redo here at the lab. The interior material, which is involved with absorbing salts and excreting material that then goes into the urine. Is that correct?
Yeah.
That's already fairly easy. And what do you do just to create those? You treat the stem cell with particular chemicals or molecules?
Yeah, so the whole process starts with a sequential addition of different molecules that provide different signals at the right stages. So basically groups well before us, I think the earliest one that we were looking at was around 2012, 2013, had decided and figured out from development of mouse and other organisms what the signals were that were important to make these stem cells turn into the right conglomerate of cells that can self-organize into a kidney specifically. And so we have utilized their knowledge to be able to pull those particular molecules and give them one or two at a time at the right time to make them turn into the cells we want them to be. And then after 14 days of this, or maybe 12, we let them do everything on their own for another 16 days. And we don't do anything other than give them fresh nutrients, which I think is kind of the miracle. They're able to do all of this communication once we give them a jump start.
And now you produced this three-dimensional image with one of our confocal imagers, microscopy, less than a month ago, I think, around
a month ago.
You were pleased when I first saw you
Yeah.
with this. Why were you pleased with what you saw here?
I mean, look at this. No, I mean, I think it was validation for me with everything that I learned from the group that's been working in Hanover on this project for a while. Yulia Keon has been pioneering this over in Hanover. It's validation for me that we can at least start to think about the next steps and that we can actually do something. I think it's a pretty cool opportunity.
And so what you'll hope to see in the years to come is the green vasculature, the vessels,
Yeah.
actually spearing into or
Yeah.
going into.
So where they're laying on top right now, I would love to be able to see them start to go in and weave themselves into little balls of microvasculature that we would have in our own kidneys. And so hopefully that's what we see.
And a change is also, using organoids. We are on the one hand, what Cory described, basic researchers. We try to understand these principles, but at the same time, we are engineering. I mean, we do tissue engineering. We want this after our, whatever we do, to look like a human kidney. So we're using both. We're trying to understand what's going on, and then we are trying to manipulate this into the right direction. And this is the international approach. This is why we read then literature, because somewhere out there in Samoa might have added a new factor, and the factor pushes this into the right direction. And then we're using the same, and then we're publishing ours, hopefully before we patent it, and then we publish. And so we are moving together this whole thing towards a growing kidney. This is more like the Apollo program. I mean, we know where we want to get to, but you all know you need tiny valves and you need tiny this and that in order to make this all happen and have a liftoff.
Dr. Haller, just today, there was news that the FDA is now considering whether to allow someone's cell cultured meat, chicken meat, to be allowed into the marketplace. It seems like, lab built tissue is popping up all over the place. The interior of this creation was generated 10 years ago, the basic principles, I think you were saying. Why is vasculature? You've been doing this for so long. You're an expert in the field.
Yeah, I mean,
why hasn't happened yet?
Yeah. So the fascination out there for organoids is huge. Pharmaceutical companies are jumping on the bandwagon. The FDA actually, after they were very cautious with new models, all of a sudden, this is all the rage. However, one of the major problems is that you have these stem cells, and the stem cells don't make a vasculature by themselves. That's very important. This is why our work is so important. So you have these stem cells growing, and after a couple of days, they make something like a vasculature, but then you don't have blood flow. So then the organoids think, okay, it's not worth making blood vessels as long as we don't have blood flow. And then the endothelial cells, which make the blood vessels, disappear. So this is why this is so striking, that Cory told them and made them to make blood vessels even without blood flow. And the wonderful thing here is that you can actually see here that there is a lumen in there. So for basic researchers, that we are able to convince these cells to form a lumen even before there is blood flow, that's what's so fascinating about our work, because without blood flow, there will be no organ. And the next step is, and we're working on that here also, that we hook up these vasculature to a preformed microcirculatory network. So we actually have the apparatus, which is not shown here, where we can have vasculature, which we have already fabricated, also from your, stem cells. And now we want to hook this up so that we have one machine where we have perfusion hooked up to these organoids and bring this together. So as I said, we are more, if you ask me about where do we stand, it's about around 1925. The brothers Wright have flown their plane in 1908, and we are looking forward to 1935, where the question was, what do you wear when you fly a plane, and not so much how to build a plane.
I want to make sure I understood you there, Dr. Haller. You said, first of all, you said lumen? What were you referring to?
Well, I mean, you know, blood vessel is a tube. So this is all about tube formation, and we are so terribly proud that we have convinced these cells to make a tube for the first time ever in an organ.
So did I misunderstand you then? Did you say lumen or did you say?
Tube. I mean, the
Tubular.
lumen is basically, you know, where the blood flows inside the blood vessel.
Okay,
So
thank
that's
you.
the lumen, sorry.
Got it.
It's like
And-
the inside of a pipe.
Thank you.
We hear that again? What was that? Like the inside of a pipe. Who was that?
Dr.
Dr.
Besser, I'm a nephrologist.
Thank you. You come to us from the skies here. So, and it sounds like you just said that you've already created the ability to get to infuse organic systems with blood using stem cell structures. Is that correct?
No, not
Okay.
yet. We know how to build a network of blood vessels from stem cells artificially, and we can induce these, what you see here. And now we have to bring these two systems together. That will be one of the really adventures of Cory's work. And he's using then our animal models for that, because we have a wonderful animal model where we can actually watch in our small zebrafish embryos how this happens. Because in the embryos, we have one glomerulus forming in the kidney, and we have a blood vessel growing out from the aorta towards that. And this is what we are watching under the microscope. And then we use knockdown methods in order to find the genes which are regulating to make this connection.
That's one of the next experiments you'll be working on or something.
Oh, he's already
so-
microinjecting.
Yeah, so I've already started to kind of work on this, some preliminary things. Haven't actually injected the cells yet, but the idea is to take, and just to give a little background, what a lot of people have been doing is to take kidney organoids and put them into mice or into chicken embryos and try to understand how those tubes from the embryos that they're being, or the hosts that these organoids are being put into, actually interconnect and give the organoid tissue life. And so the idea is that we would be able to do this in a zebrafish and watch the process happen in real time. But we're still learning how to get that done properly. So it's going to be a little bit.
And what zebrafish is becoming the nephrology of the lab to some degree as our axolotl, but we're, you know, zebrafish is the thing we do here. And Dr. Drummond, of course, as well as Dr. Haller, have been working with him for a long time. What do we get out of working with the zebrafish as opposed to a mouse model or other models?
That's exactly what you were just saying. So they're transparent.
You
Especially,
can watch them, you know, you can actually, we can invite you to come down to the lab. We actually can show you how these blood vessels grow from the aorta into one kidney. And that's unique. In a mouse, you bring this into a kidney, then you close the mouse, and then you have to open it again and do histology and all that. But we can actually, if we are successful, we can actually watch this under the microscope, and that's the beauty of our model.
What other experiments are you planning, let's say in the near future? Maybe at the end, I'll ask you like your dreams, but what kind of work can you talk about at this point that you're planning?
so we just did a first pass with a molecule that I found really interesting. So we all know the stress hormone cortisol, right? I probably had a little bit coming up here to talk to all of you guys, and it's calmed down a little bit now. But I think what's really cool is we know that it's based, at least on normal rhythms, through light. So when we wake up in the morning, we get a spike of cortisol, and the light that shines into our eyes produces that spike of cortisol. But it's also really interesting because it helps regulate embryonic development. And it's highly controlled by the placenta and then within the fetus, which I think is fascinating. And I found this doing just some random reading of literature on cortisol, and I thought it was kind of something to pursue. So I looked around the lab and I found some corticosteroids, and I decided we might as well try them out at the right time, right? So I've mentioned that it's sequential, and when you put these molecules onto the organoids, it matters. So I thought I would try it out. And Roy, if we want to move to the next slide. So I think what's cool about this, and I'll stand up, is this is a control culture. So this is at 9 days, what our cultures look like when they're happy and healthy, which is pretty good. You can start to see some tubes forming, and that's nice. And then this is without the treatment that we had done with the 3D image. And then I'll just skip to this one, but this is when I gave hydrocortisone for a certain period of time over the course of, say, 6 days, at the same time every day. And you can already see differences in what these organoids are starting to look like. And I think that's pretty fascinating. I don't have anything beyond that because this just got done about a week ago. And so the nice thing is, if you move to the next slide, you can also see a dramatic difference in overall the tubes. And I think this is really fascinating. So it's harder to see in this culture, but you can see some tubes interconnecting with these little dark spots, which if you look really, really closely, are those sets of tubes that look like this up here. But then the nice part was we used a molecule called VEGF, which is important for blood vessels to stay healthy. And it looks like that having both of those molecules together during this time has almost stabilized what looked to me like maybe blood vessels, but I don't know yet.
So is this related to like biorhythms, diurnal cycles? Is that a separate experiment?
That was how this got started, but I'm not 100% sure that that's exactly what's going on. So the hypothesis was that inducing circadian rhythms in these cells using exogenous signals might produce. Honestly, I didn't think about tubes. I was really thinking about the kidney and seeing whether or not it developed faster.
So we have examples in the human body for that. We haven't talked about that, but
Yeah.
there is a tiny gland, the pineal gland, which regulates a lot of how you feel and what's happening. And this gland has a 24-hour rhythm where it makes new blood vessels and abolishes the blood vessels within 24 hours. So at a certain time of the day, you have new blood vessels sprouting in the pineal gland, and at certain times they disappear and they change their behavior, the endothelial cells. So there is a lot about circadian rhythm. Nonetheless, when Cory first showed me the experiments, I said, great, wonderful. What's your hypothesis?
If I can make comment, there's more than one rhythm in the body. There's a circadian rhythm. There's also a 7-day rhythm, which is important for some certain disease states or the development states. So there are multiple, and I'm wondering in the embryogenesis whether some of these longer rhythms, which aren't daily, won't be important as you try to develop your model.
Well, this is the beauty of the doing basic research as we do here. You know, Dr. Haller talks about getting a patent and winning the race and that sort of thing. That's really, and he knows it, it's only half the story or less here. Being able to just think up questions in the comfort of this place, I think is really, really valuable because it produces the kinds of moments in science that you may not have expected. Jerry, you're looking at me like you're waiting for something, so.
Hitting on point, I just wanted to emphasize, I don't know if it's fun, but
It's not working. Roy, can you activate that?
There we go. So I just want to go back, Cory, because I think it's such an important point to illustrate for the audience about how the scientific process happens, right? Because when I think about what you're trying to do. I mean, to really oversimplify it, it's like you're trying to bake a cake without any recipe whatsoever. So walk us through how you know what questions to ask. I mean, how did you think about circadian rhythm? I mean, you mentioned you were talking about hydrocortisone, but cortisol, but I mean, to me, that's so fascinating that you just happened to land on something that you know, was there reading and a lot of other thought, obviously there was a lot of thought process that went into it, but I'm just curious because it's that creative thinking that really leads to some amazing or interesting results.
Yeah. No, that's a great question. So I did my doctoral thesis in yeast, which is very different than what we're doing now. But what we were looking at is how cells change their shape. And one of the interesting things that we found is part of that is regulated by the cell cycle, so cell division. And when I think of cell division, I think of synchronicity between, you know, the signaling inside the cell, but I also think of the external factors. And one of the external factors in humans that we lose in the dish, more or less, is circadian rhythm. And so I started getting kind of interested in this. And then at a meeting, one of our other faculty here, Jim Kaufman, gave a talk, and he has some previous, you know, background looking at circadian rhythm and cortisol, and just sparked that thought again. So I sat down with him and asked him if he thought this was an interesting question to look at circadian rhythm or circadian process in cells and how I might go about doing that. And he suggested something along these lines. So then I did a little bit of reading just to clarify what I wanted to do and understand if it was even worth investigating. And yeah.
So cortisol is actually a stand-in in the lab for the Earth circling the sun, or actually the Earth spinning on its axis.
Yeah, the idea was to try and mimic it using this 24-hour cycle that we were talking about before.
And now it becomes complicated. We can ask you, you know, imagine you're running a lab, you're running a program. I mean, you have quite a few people. And now Cory with Serendipity has started all of a sudden circadian rhythm in organoids. And I can tell you, not a lot of people before that have thought about that. So that's very original. So would you now embark on circadian rhythm with a full program and look into all the genes and the approach? Or would you say, well, that's enough. We have shown that this works, but we are concentrating on other things. So we either miss out on the Nobel Prize, or we are spending a hell of a lot of money on some weird idea which will lead us into a jungle of nowhere. And that's what research is all about. So sometimes you make the right turn, and sometimes you don't.
Mm-hmm.
I think all of us would enjoy learning a lot more of the details around the work both of you were doing. I did want to raise, before we open up for questions a little bit, although there's one back here if you want to go ahead at this moment, or were you stretching? Oh, he's which is whether there are ethical issues that we need. It's 1925 in organoid development. What should we be looking to understand about the ethics involved here? And as I've tried to think about what those issues might be, you know, the first one that I might be natural to land on is access. Like as therapies related to organoid development become available, maybe cystic fibrosis provides an example here. What do we need to think about who gets access to what probably will, at least initially, be pretty expensive therapies? And what are the other ethical issues? You know, one thinks of, you know, sort of sci-fi stuff where we're creating normal human beings with organoids. But what should we be thinking about? What do you guys thought about?
Well, if I may answer, you know, from the point of a nephrologist, you know, the ethical issues at the moment with stem cells, there are none. I mean, we had the problem with the embryonic stem cells, and now we can work with these cells, so there are no ethical issues implied at the moment. However, when you think in the future, we are able to generate a kidney, and perhaps you can buy a kidney, and if you need one, and so then it becomes ethical. Just to remind you, it's always about ethical problems. When dialysis was introduced in this country and in Germany, some of you may remember, the act
Holland,
was only for people
yeah,
the age of 40. If you were below 40, sorry about that. When you were over 40, you didn't have access to dialysis. This was 50 years ago. 50 years ago, imagine that. When I tell these medical students today about the ethical problems of access to dialysis, nobody, they think actually, they don't believe me. And this will be the same now. We have therapies at the moment. I've been involved in new therapies and they cost $1 million per year. So who gets access to that? So I think the accessibility of organs, that will be an issue. At the moment, we have already discussions about that, not serious discussions, but hopefully the price for a kidney will be in the range of a brand new Cadillac, which would make it then at least affordable. So that's the ethical issues. Producing a new human, the Jackson Laboratory is also using organoids and they are working on brain. But here these are very crude neurons and endothelial cells, as our cells are very crude kidneys at the moment. So then when we talk about rhythms and we talk self-organization, for me personally, when we do research like this, it's always that you have this feeling like you're being in a church. I mean, you are dealing with mechanisms, not the cell, which mechanisms which are so complicated and so difficult to understand that you stand in awe when you look at your experiments that somebody has really thought about this whole concept and brought it to work.
Cory, have you thought about some of these things?
Oh, absolutely. And, I think the nice thing is that, as we were saying before, the stem cell issue is kind of a moot point at this point because we're not getting them from embryos anymore, which is great. But I think something really interesting popped up in the last 2 weeks where some preprint papers just went up from 4 labs. I think this was a week ago, all on Thursday and Friday. I think it was two papers a day. And they're growing actual embryonic tissue from stem cells, which is fascinating to me. That's true cloning in a sense. But the nice thing is we have rules around how long this can go and to what stages they can go. So I think, in terms of ethical with the full human, there's a lot of discussions that need to happen. But I think that, really it comes down to with kidneys and donor tissue, it is absolutely accessibility.
And I'm wondering about, are there questions to be asked about donor compensation? I mean, I guess if you're making, if I make a personal biobank from my own stem cells, because I can afford to, that's a nice thing. But is there a question about if you make something that's useful for, let's say, testing drugs from a stem cell that you bought off the shelf, who gets paid? How do you adjudicate questions like that?
Oh, well, you actually, if I may answer that, when you enter a hospital, you sign all the papers so that the hospital or the scientific institution can use your cells for that. And it's also now clearly stated about compensation. So that's already now part. There is, as you know, there was a famous story about HeLa cells, one donor, and then the cells were used all over the planet and no compensation whatsoever. So this is taken care of. To be honest, at the moment, there is not that much coming out of it. So the examples I described is you get your personal compensation because your tissue is used when you have the disease and drugs are tested for that. At the moment, we are a far cry from that. And as I said, it's very personalized medicine. So that's also beautiful. You will not be able to give us your cells and then we make a kidney and sell it. It can only go back to you.
I want to encourage people to think about questions If both folks watching on Zoom, as well as people in the audience here, if anybody has something popping up right now, it's be good to get the ball rolling. Jerry?
Sort of related question, but sort of the
Yes, sir.
Colleague. Sorry about that.
I'm curious on the ethics side, sort of related to this topic, but tell us a little bit about kind of what's happening in Europe right now in terms of use of animal models and how you see use of organoids maybe moving forward there and where the US is in that
Yeah.
sort of transition.
That's I mean, that's really something very important. And this has to do with research and which models you use. I think you're all aware that the world has changed dramatically. When I was a medical student, we had dogs and we had animal models with much larger animals. That's totally out of the question to use this. Imagine, I remember 30 beagles in the lab, and we are experimenting on these animals. Everybody is shaking their heads. You know, that's impossible. Now, this is now rapidly moving into rats and mice. Rats, not so much, because rats don't have a lot of friends, but mice have a lot of friends. So actually, the European Union, and this gives you a glimpse what's happening, the first discussion, official discussion about banning all mouse experiments completely. You can imagine this, not only the tax is out of business, but for the research, it's complicated. And we are already experienced this. When I write a grant and there is a mouse experiment in the grant, it takes 1.5 years in Europe and 9 months in the US to get this approved by the ethical committee. So these are now real ethical problems, which animal model you can use. At the moment, our zebrafish are out of the equation, so to speak, because they only become animals after the embryonic state. So we are very grateful for that. However, with the organoids, everybody thinks that's the escape. You know, you can use these organoids and then we don't have to worry about animal models anymore. And this is why pharmaceutical industry, this is why the FDA is so interested. We will have next year, most likely, a meeting with the FDA about models to be used in pharmaceutical development. However, I think you have learned this from Cory, how complicated this is. To be honest, we are not there yet. And we are very cautious in the lab not to raise false hopes. Yes, we want to have a kidney. Yes, we want to have renal tissue to do and develop new medications. But at the moment, we are like in kindergarten. We are playing with that. It goes dramatically fast because such a lot of people are working together. But these are the real ethical issues at the moment that we have high hopes, but we are not there yet.
I'm curious, is this one of the, I've heard Jim Strickland of MDI Bioscience, a subsidiary, talk about this, about the sort of midpoint that maybe some of our models, such as zebrafish, could play between cell culture and mice, and an important one that is more robust for toxicology and other kinds of testings or efficacy testing than in silico would be, because you have a whole organ system that you're running your test molecule through. Do organoids potentially play that role? I understand what you're saying, that it's not there yet because we still need to learn how to make it.
Yeah,
but
I mean, in a nutshell, what we are trying to convince, the checks was very successful to convince everybody on the planet that a mouse is basically a small human being. And we are working on the concept that the zebrafish is also a small human being. And I can see your response. We have to work on that, but basically it's true. I mean, the zebrafish has all the components. I mean, a kidney, a brain, a heart, vasculature, and when we look under the microscope, they look the same as ours. So it's always the question about the model, and we have a hard time at MDIBL and others also, then to make this point go from use as zebrafish now, but eventually it will be all stem cells.
And very interestingly, I think around this zebrafish is they are so good at regenerating their kidneys, they might be able to show us something that mice couldn't. around our own abilities.
Yes, they are more advanced.
Brett, I want to just interrupt you because you got a couple questions.
Yes,
there.
Anna, you have some. Please.
So if you want to take yourselves off mute, you can ask Dr. Bessarab. Do you want to ask your question?
I had to make a comment. This is wonderful work and I wish you a great deal of success. But you have a long, long way to go to actually get a functioning kidney with all the structures, because there's a variety of cells within just the tubule, let alone the glomerulus, which is ultimate. That's what you're starting with. You got to get that tubule to wrap around it, and connect to the vascular structure. So I encourage you to do that. But I think people are prematurely worried about the ethical question that's going on until you get more specificity in terms of the function, so you can look at the toxicity on tubular cells or glomerular possibilities. You're years away. So I think we should not forget the ethical concerns, but I think they're disproportionately put out of place. I remember the day when I operated on sheep in my lab and dogs. And now it's hard to even get a rat experiment done. And so I retired from basic science.
Yeah.
I've left a whole bunch of comments in the chat that you're welcome to read.
I mean, just one remark you made is very important. I strongly believe in function. So to understand all the molecular details, that's too much. But if we have structures and then we tell our kidney we want to have what the kidney should do, then I trust the cells very much that they do the right thing. So we have to start blood flow, and then we have to start urine flow, and then let them do their job. And that's the problem we're facing, how to start the blood flow. And then they have inborn ways how to develop themselves. So I'm hopeful that when we use the intelligence of our cells, that we'll get there.
I think we have more questions from our Zoom audience, and maybe one more from Zoom, and then we'll come to you in the audience.
Chuck, you want to go ahead?
Thanks, Jerry, and I'm really appreciating the presentation. Let me go back to the beautiful picture that and video that you produced in the beginning. It was reminiscent from the earlier developmental embryology days of the epithelial mesenchymal interactions that we used to talk about. You have an endothelium surrounding a putative potential nephron. To what extent do you need to get them face to face for some morphogenetic tissue interactions that may be at the cell surface? Cell surface markers, are they available to you? Can you push them a little closer to get them to talk nicely with each other? Something of that nature?
Yeah, that's a great question. So I think that's where we need the work, right? So in order to understand the functional characteristics of these tissues and whether or not they express those, we do need to understand each cell in itself. So the nice thing is that recently, in the last several years, there's been an explosion of single cell sequencing work that you can learn important transcriptional gene expression programs that regulate these things, and then you can kind of infer the outcome of that. And in particular with this image, it's how do we quantify those functional outcomes? And so I think those are also parts of what we're trying to do. And again, just each color here is representative of some of these, functional markers of the different cell types. So it's slowly getting there. And other groups have some different markers that they're able to use. But yeah, I think that's ultimately the question we're trying to answer is how can we understand functionally and where we are on that functional level.
We had a question here in the Marin Auditorium. Jerry, maybe if you, the mic is at the back of the room here.
The cortisol issue is, as I understand it, the stress component of cortisol affecting your ability to use it. And I look at collagen organization and nanostructures,
Mm.
and I look at stressing bone tissue and stressing it too much. What do you get? Disappears.
Yeah.
You stress it right, you got it. So what is that number? Who knows? But is there any research being done on magnetism and of tissue, positive, negative, and growing tissue in the realm of that, along with nanostructures and scaffolds that are generated quickly in the human body? Because we're really talking about nano holes or nanopores for a place to align those cells and to form what's the miracle. And that's the juice that we're looking for. So I think that it's in a combination of biochemistry, but it's also mechanical properties of the elements. So we need the brain power that's going on in the research areas of beyond the biochemists, the mechanical nanopore people like Oxford Nanopore and companies like that are making products that do all the gene sequencing.
Mhm.
And I think that's where your answers might be.
We fully agree. I mean, we have not been talking about that. We only mentioned our microvascular system that's bioprinted. So that's silicon material, that's nanotechnology. So these small blood vessels where we grow the cells in, and that was what you were mentioning, this is engineered. And we do bioprinting. Some groups use bioprinting for these tubular structures or for the vascular structures. We have research groups using silk, for instance, which is organic material, and then you can use this as a scaffold. So the scaffolding is a serious problem, but also a big promise, because you see here these cells, and we have talked about the tubes, we have talked about the lumen. Now the big question is what's happening out there? Do they actually make their own scaffold? And this is what we are looking at the moment. And then when you print these things and then you grow them in there, then you may get more stable blood vessels. And then you can print with biomolecules in the printing material, thereby mimicking the embryo and things like that. And these are also approaches, so we can't do everything in our lab. As I said before, this is about international collaboration. We have our focus, but we need the interaction with the international world to move this whole thing forward.
That's fantastic.
So Jerry, have we taken care of the questions they have answered? Should we have time for one more or two more?
Question. It's kind of on the ethical subject. When, if ever, does an institutional review board come into play here, and what would constitute a reasonable institutional review board?
I think you might be better at answering that because I've never had to work with one.
Yeah. At the moment, it's only about cell culture. So there is no review board. We have for our animal experiments, we have institutional reviews, and we have standards in general. But for this work, at the moment, there is no review work. It changes immediately when you use it for pharmaceutical research. You may be aware that we have built one lab down there at the water where we fulfill all the criteria for institutional review, producing data for pharmaceutical industry. So MDI-BL is also, although we don't look at, but we are moving into this area that at the moment for organoids, no institutional review necessary.
And we have one more question, then we should wrap it up.
Can you describe generally what the four magic drops are that convert any kind of a fibroblast adult cell into a stem cell? That's one question. And secondly, how do they deal with telomere reduction and finite lifetime in that process?
These are both very excellent questions. I mean, the first question should go to you, and then we have an exam here. What do Yamanaki factors actually do?
Yeah, no, that's great. So I think the interesting thing to that is that there's really one or two that do the job. So I believe, if I'm remembering correctly, there's one called KLF, and then one that is OCT3/4. Those two factors produce the gene expression programs that regulate kind of pluripotency, the meaning where we have, different cell lineages that can be made. So they turn directed differentiation off and restart the programs that maintain this kind of, what's the word I'm trying to think of? Stem cell-like phenotype. And in addition to those two, there are two other factors that boost or enhance that transcriptional expression. And so those four droplets really are they can change.
And I'll take the second question because it's a very important one. You all know that embryo does not mean growth, but it means also that cells are dying all the time. So you have regulated cell death. And these structures here are growing, and at the same time they're dying. So the whole atmosphere here is one of inflammation. So this is not just one ball growing, but structures, and they build and then they fall apart and then they regrow. And that's what the embryo is doing. We learned that the hard way, because when we first brought cells from the outside into this, they burned. They became inflammatory cells immediately. So we realized that there is an inflammation burning in these organoids. And one reason the cortisone may have worked is because it brings down the inflammatory response, and all of a sudden the embryo becomes more stable. So, but it's a very important question. The apoptosis and the rate of decay in the embryo and the embryos. And then all of a sudden, we have spent now an hour and 5 minutes here. It tells you how damn complicated the whole thing is.
But you can see some of the directions where it's going to take people in their fascination with these complicated issues. Dr. Haller, Dr. Johnson, thank you so much. Jerry Bowers, thank you so much. If any of you, I think we have some refreshments, and have more questions, you'll have an opportunity. People on Zoom, you can email communications@mdibl.org for any follow-up questions. We'll be sure to get them to Cory, to Dr. Haller, and I'll let Jerry finish things off.
Yeah, well, before we let Cory go and let his cortisol levels go actually back to normal, I can't miss this opportunity to just ask you, when you were on the ski slopes in Colorado, did you ever think that this is where you would end up and that you would have this international sort of experience being here in Maine?
No, I thought I was going to be internationally skiing, but yeah.
But seriously, I mean, one of the things that we are really trying to accomplish here at the lab, I mean, in addition to the fascinating work that's happening, I mean, I hope we gave you a good glimpse into some of the exciting work that we're doing, but it's also about careers for Mainers. As a native Mainer, I'm pretty excited about that kind of stuff, and I know Cory is too. But it's really, it's an international program. Yes, we have some wonderful, wonderful folks from Maine, but we also bring students here from all over the world. And so this is another really important part of the work that we're doing here at the lab. And for our Star Point Society members, we'll have a gathering on Wednesday where we'll get a chance to hear from some of those students and learn more about their personal stories and more about the role that they are playing in work like what Cory just described to you. So if you're interested in learning more about those kinds of programs, please come see me. I'd be more than happy to tell you a bit about that. And I also want to put a plug in for some of the many events that we have going on this summer. As you may have heard, it's our 125th anniversary. And so we have some special programming put together. Our next Science Cafe is on July 10th. And I think we'll be, I'm not sure if we're back here or if we're in MCBI, but we're here in Marin Auditorium. So this again is a great glimpse into some of really the future direction of science. So we're talking about multi-omic maps. Don't ask me because I can't tell you. But what I do know is that it is incredibly exciting work that's taking basically bioinformatics and proteomics and all of these different things and pulling them together into one interesting research approach. And this, again, is a great example of the international scope of MDIBL because it's featuring two scientists from Aachen, Germany, who are leaders in this arena, who are really kind of forging new ground in this approach in science. So we encourage you to tune in and continue to join us for these science cafes. And as Fred mentioned, if you have ideas or interests about other future topics, please let us know. And one final plug, on July 12th, we will have our family science night. So if you have little ones or you just want to pretend that you have little ones and come for the ice cream truck, I hope you will all join us. It's from 4 to 6, and basically we turn the lower campus into an outdoor science fair, and all of our student fellows that are with us for the summer put their work into terms that young folks can understand and get excited about. So it's just a great atmosphere, and I hope you'll join us. We're going to have some birthday cake and cupcakes, and you never know what you're going to get if you come here. So
One
Yeah.
short remark, tomorrow evening, 5 o'clock on stage, you, together with Dave Evans.
oh God, that's right. Yeah, the
now
library, I
in-
forgot.
In the library, and you can learn everything you want to know about the history of the lab and
right.
important people.
That's right. I totally forgot. I think I was maybe just pretending that it wasn't.
Okay.
So we're going to be at the Southwest Harbor Library tomorrow, and these are a series of library talks that Mike Hayes organized for us. We did one at the Jessup last week, and as many of you know, Dr. Evans put together just a wonderful, wonderful volume about the history of the MDI Biological Lab. So he is the true expert. I'm kind of facilitating the conversation, and it is a fun way to learn a little bit more about some of the amazing science that's been done here over the last 125 years. So there are 4 talks altogether. We'll then be at the Bass Harbor Library and then at the Northeast Harbor Library. So those dates are on our website if you're interested in the future dates. But anyway, thank you all for joining us. I'm sorry that we kept you over time, but the subject was worthy, I think. So thank you very much.